Alright, so let's start up for today then. The map for the week is shorter than usual, as you know, I hope. We don't have class excuse me, on the 16th, which is Friday, but please note that there is a quiz that is officially due then. If you need to go away, please submit before then. We've covered all the material, as far as I know, that's going to be on that quiz, or will be at the end of it as of today. You'll have plenty of time to get that done, but the date that's listed in the syllabus is still totally true and right there. And today we're in carbohydrates for the last time. This time talking about bigger ones than we've dealt with in the past. It was only up to 2 as of Friday, but now here we're looking at so-called polysaccharides. And monosaccharides are the monomers here. In this case, as we move up into this size region, we're by definition talking about more than 2, so at least 3 and usually much bigger than that. And like this says here, we're looking at only one kind of monomer, and that's D-glucopyranose. Okay, so you need to think of this whole thing that we assembled as a 6-membered ring before. Now you're going to view that as just a monomer in something much bigger. It's a polymer, of course, when it's bigger. And this thing right here, please remember, is honest to goodness glucose, but it's in its ring form. The polymers that we'll be talking about from here on are made of a bunch of rings joined together. All of them glucose. There are other naturally occurring things, but we're leaving them out. All right, we talk about them several different ways. One of them is the monomer. There's only one. The kind of glycosidic linkage, and remember this right here. They're going to be either alpha or beta, and that's talking about the linkage on carbon number 1. So we did some practice with that last time. We'll do some more this time. Branching is something to be worried about for the first time as we move up into this zone, this size zone. What we mean by branching is that it's going to be off the main chain, if it's present, of the polymer. Some of the polysaccharides are branched a lot, some of them are not branched much, and they can branch from different points. We'll get into that. All right, well, we're headed now into, this is your little roadmap, 3 different polymers of glucose. And remember, glucose right here, again, is going to be the only monomer that we'll consider. It's important because it's the source and the monomer in your most important polysaccharides. You need to be able to describe them in words if you're given a structure, and if you're given a drawing, be able to recognize it and say something accurate about it. Okay, so here's your first version, your first polysaccharide. Common name of starch, you've heard of this before. So let's just say where we are on the map right here. This is polymer number 1. And this is a storage form. The plant thinks that it's storing energy for itself, except of course we come along and harvest the plants and it becomes nutrition for us. But the reason that any plant, potatoes, corn, what else is really starchy? Rice has got a fair amount, right? Those of you in dietetics. Okay. Any starchy plant thinks that it's making this for itself. And it does work that way. But we come along and we as humans are able to digest it. All right. Now there's more than one thing you can say about branching here. There's branching in starch. It is branched. So is this one unbranched? No, at least not that we're going to consider. So the polymer backbone, you need to be able to say this about it, is so-called alpha 1,4. I'll put some labels on that. And the branch points are alpha 1,6. Now, if you're wanting to make a difference between these 2 main parts of the diagram right here, think about this as kind of level number 1. So this is now drawn as just the backbone. Now, real starch has some branches off of it, but we got to start somewhere, and this is what it looks like. So this 1,4, let's take this apart and start to connect this label right here to the structure. Alpha 1 and 6. Okay, in ring number 1, hope you're starting to recognize where carbons are. There's number 1, and so that would make this one number 4 over here, and that's the first ring. There are other carbons, let's ignore them for the time being. Then in the next ring, same kind of thing. There's a 1 and there's a 4 right there. Okay, and we're talking about this business of alpha 1,4. Okay, now remember what you're talking about with that kind of label. Alpha means, and I'll just put this little label on it, alpha means down. And that's on the anomeric carbon, which is carbon number 1 on that left-hand ring. And so that's where the alpha comes from. This is also down. So down also on this anomeric carbon. It's like that for all of them. This bond right here that's left blank, that goes to the rest of the molecule, and there's a lot of it. And on this end, same thing. All right, that's the unit. That's only 2 glucose rings out of hundreds or thousands, and that's how the backbone itself is held together. Alpha 1,4. This other version that you're looking at now is stepping it up a bit. Let's see if I can get all that on screen. So this now shows a branch point. Now you may remember from the previous class, what we got to is the point that any of these OH groups can be a hooking on point for anything else. And this is indeed what happens for starch. So just to make sure that we're separating the linkages correctly, let's say here's on this ring right here, here's a 1, and here's a 4 on the next ring, and that's alpha. Okay, so once again, alpha 1 to 4. I'm going to keep that small on this version of the drawing just so we can concentrate on what's different here. Now, if you come up on this ring right here, let me get all the numbers on it. 1, 2, and 3 are around that direction. We've already put on 4. 5, and don't forget 6 is actually part of that first glucose unit. Number 6 always dangles off the edge. And so now we're looking at a linkage to a carbon number 6, and this one is exaggerated, of course, to make it all line up here. But again, this linkage is alpha, and that's because the oxygen points down. And up on this ring right here, the third one that we're joining to the main structure, you're looking at a 1 and 2 and so forth. It's 1, 6. So the branch is what we're talking about in this zone right here. This whole thing right here is from carbon 1 on the first ring to carbon 6 on the main chain. So you call this linkage, all that information put together. This one is alpha, and I'm going to ask you to tell me what the 2 numbers should be, and I'll put them in a different color. Look at your first ring, then look at your second, and you ought to be able to get a number as the address. So what I put for the first ring? It is 1, and how about for the next one? 6. All right, down here in the main chain, we're talking about alpha 1,4. That strings everything in a nice long line. Alpha 1,6 is where the branch takes off. Okay, now again, you're looking at the rest of the molecule being somewhere else, but look what's going to be different this time. Now you have a new point, and this is up on the ring that's bound through its carbon number 1 to 6 down here. Now you've got 3 different zones where the molecule can grow. And that is one thing that makes starch decently easy to metabolize if you're a human being. You have starch metabolizing enzymes that know how to recognize branch points. It's like, aha, I know how to tear this molecule apart. I'm going to start with the branch point, and that'll make the whole thing easy to digest. And that is indeed what happens. Okay, so that's kind of polymer number 1. Now there's another option, which is glycogen. So maybe make sure that you call this polymer number 2. And what is not different between glycogen and starch is the monomer. Both of them are made out of glucose. So that much you can expect to be the same. And let's just kind of go into this as it is. The main structural difference that you'll want to think about is that it's more branched than starch, and for that reason it's quicker to metabolize. Alright, which is fine. Again, the polymer backbone is alpha 1,4 and the branch points are 1,6. So what's the difference between the two of these? I won't put as much labeling on this time, but let's make sure that we get kind of the big picture right in the comparison. This version right here is the backbone with the branches not shown. This right here is both the backbone and a branch. So kind of the same as on the other page. Just looking at that comparison, you couldn't tell whether you're looking at starch or glycogen. What you have to know is you have to be told, or it has to be drawn or something like that. In this case, though, there are more of these. In glycogen. All right, we'll get to the metabolic side of this, almost physiology, as we go farther on in the course, but the structures we're talking about make it look like starch and glycogen are a whole lot alike, which they are. It's just that there's a lot more branching in glycogen. So maybe here's one more chance to look at labeling. This again is alpha 1 to 4. This linkage right here is alpha 1 to 6. This one is alpha 1 to 4. All right, that's the whole difference to the extent that we need to be worried with it right now. All right, please note, once again, humans can and do metabolize this one. That's true of both starch and glycogen. Oh my, is it true of starch. A lot of calories packed into starchy foods, right? At least that's the image. Okay, that's the first two, very similar to each other, and the world changes a lot with this structure right here. So cellulose, consider that number 3. Polymer number 3. This one is unbranched, and so I hope that you can kind of remember that by looking at this structure right here. If you've shown this on an exam or something like that, it'll look pretty straight like this. It's both drawn that way, and it's called that, and there's an immediate consequence of that. It's harder to attack by enzymes. And by the way, we don't have the enzymes to do that. Humans cannot digest this. Now, there are organisms that have enzymes that destroy cellulose with no problem whatsoever. Termites are an unfortunate case. Right? If you get them into the wood, in your walls, just watch them digest cellulose. They're extremely good at it. And the polymer backbone here is the other main difference. It's beta 1,4. So let's talk about where that comes from. Here's a 1 and here's a 4 on the first set of rings right here. Here's the rest of the molecule, here's the rest of the molecule in that direction. Okay, now what you want to look at here is this oxygen right here. This oxygen points up, so that makes it above the ring, it's beta. And the linkage is now, it's still 1,4, but it's not alpha anymore. This is beta. Carbon number 1 on one ring to 4 on the other ring. Okay, now just I hope not a trick question, but a review kind of question to make sure you're kind of keeping all this straight. Why is it that, well, you kind of see why oxygen needs to point up to fit the term beta, right? That's a given. But look over here on this one right here. Sorry, we're missing a line here. On number 4, we've got the assumption that this oxygen, which is now partnered to both of these carbons, this one we're saying that one has to point down, but we're not putting any special letter on it. Right? The beta only refers to what happens on that first carbon right there. It has to point up because there are two options. Why don't we say something like alpha or beta about number 4? Yeah, that's a way to kind of probe and make sure you know what you're talking about here. Okay. Okay, it's stereochemistry, right? If you drew the oxygen on number 4 pointing up, it wouldn't be glucose anymore. It would be one of the other stereoisomers. There, in the world of D stereoisomers, there are, let's see, that one can be either way. So you've got 1, 2, 3, you've got 3 chiral carbons. That one's not chiral. This one is constrained to be D. That one can be either up or down. Okay, but these remaining 3 right here, you could draw this ring correctly and monkey with 3 carbons, and you'd be changing the name of the molecule. That's how sensitive the whole thing is. Big difference to pointing an oxygen up or down, and actually that makes it simpler too, right? Because if you know where number 1 points, the others are hardwired in. For it to stay glucose, everything has to be like this. Number 2 down, number 3 up, number 4 down. Okay, that's just the way glucose is, and you can forget about all the other options. So that's 3 polymers made from glucose, and those are the ones we'll deal with in this class. There aren't any others. All right, so here's now a way we start to blur the lines between carbohydrates and proteins. It's not like nature hands us totally separate baskets and they never meet, because you have a hybrid protein carbohydrate thing, which we call glycoprotein. So the glyco part refers to a carbohydrate, and it is bound to a protein. The protein is exactly what it says. It's a polypeptide. And they are bound to each other through one functional group on each one. There are usually lots of options. Carbohydrates have all kinds of OH groups. The proteins have plenty of options, plenty of functional groups for joining up. So let's dissect this very briefly right here. Usually, if you're using this term, glycoprotein, it's not just like one little component of one and massive the other one. That sounds more like a cell membrane. But if you're talking about an honest-to-goodness molecule that's called a glycoprotein, it's usually like about 50-50, mass and mass. That's the way the term is used. And your R groups are going to link up to snag the carbohydrate. So all that put together, you'd imagine being something like this. So here comes part of the polypeptide. Here's the amide bond. And I'm drawing this as it would be kind of drawn on one of your answers to an exam question. So there's an amino acid that has partners on both ends. It's buried in the middle of the molecule somewhere. There's that much. This would be your alpha carbon, and then here's a space for an R group. All right, I'm just going to arbitrarily pick one. It would not have to be this one, but here's a serine residue. It's got an alcohol on it, and you could use that point to form a connection to a carbohydrate using exactly what we've been just drawing for the last several minutes here. So you could go like, well, here's my ring. Okay, six-membered ring would fit just fine, as it does when you're linking carbohydrates together, glucoses together to form a carbohydrate. That right there could be the start of the connection between the protein part and the carbohydrate point. Now, yeah, you need other stuff here. So again, carbon number 2 needs to have its OH down. Number 3 needs to have its OH up. Number 4 down, and number 6 needs to be sitting up here like this. Now that's just one sugar. You have your hydrogens stuck in like this. Okay, this right here, this spot, is going to hold your 2 units together. That's a way of making an honest-to-goodness covalent bond between a protein and a carbohydrate. Now, that doesn't have to be all there is to it. Okay, this piece right here could be a bigger carbohydrate. So that's one possibility. It could branch. And so forth. In the protein piece, you've got the rest of the molecule on either side. There are millions of molecules that are possible, not even all that big, that would just differ in where they attach, how big they are, what functional group holds them together, and so forth. All right, the carbohydrate part is a monosaccharide or a branched polysaccharide, usually of 2 to 10 monosaccharides. These aren't looking big things compared to enzyme molecules, but still there are lots of opportunities for sizes and varieties and so forth. And here's what they do and why we talk about them so much. The emphasis of this class, it's only a 3-credit class, is on metabolism, but we've got to think a little bit about other things that biomolecules can do, and this is a major one right here. So marking cell types is most obvious when you start talking about blood types. Okay, so different glycoproteins, what that's kind of saying is that there's one type of glycoprotein for type A blood, and there's another one, this is a different glycoprotein molecule, for type B. And it's not hard to see how it would be easy to make a big difference in a molecule with this unit to it. Just branch it a little bit more, change the polypeptide, change the attachment point. They would fold up very, very differently without changing the structure too much. Another thing that you can tell about these is they're important on cell surfaces. As a group, molecules like this tend to be parked right on the outside surface of cell membranes. Okay, meaning that if something goes wrong with the gene that's in charge of making these things right here, you may be looking at a malignant cell right away. Because if you have cells that forget how to stop growing when they come in contact with each other, you have uncontrolled cell division, right? And the definition of uncontrolled cell division is malignancy. That's one big area in cancer research, trying to figure out how to fix damaged cells so that maybe they will start recognizing each other again. If there's an enzyme that is making too many or not enough of these things right here, figure out a way to inhibit or to speed up the enzyme. It's a whole world of strategy unto itself when it comes to cancer. And then they'll also mark the age even of a normal cell. So it turns out that the fewer of these there are on even a normal cell, the more the cell is giving the signal, I've come to about the end of my regular life cycle, time to break me down, recycle all the components. As you probably know, there are cells in your body that have the full-time job of roving around and checking other cells to make sure whether they're good or not. Some cells will even, they have their own programmed self-destruction so that they sense themselves when things have gone about to the end of the life cycle, and they will very carefully disassemble themselves, called apoptosis. Have you heard of that term? Okay. This whole molecule right here, this whole family of molecules is very important to that. Instead of just, lysing the cell and busting it up and throwing all the components out into the surroundings, it's a controlled disassembly. All right, glycoproteins are important for that process. All right, so we will make the jump here into another chapter and stay on schedule pretty well as far as I can tell. This is the third of 4 major categories. There are 4 categories of biomolecules in this course. We've been through one of them all the way, proteins. The other one, the next one, the second one, carbohydrates. Here's the third one, lipids. The fourth one will come towards the end of the course when we talk about nucleic acids. This is the third major one and the last major one for now. All right, lipids are not categorized by functional group. That's probably the first thing. So they're classified, start that word again. They're classified by solubility. They are not classified by functional group. Remember how it is for proteins. Classified by functional group. You bet. They're polyamides, all of them, every last one of them. Carbohydrates, the one we dealt with, they're all either aldehydes or ketones with a lot of OH groups. Lipids can be like anything and everything as long as they dissolve in some non-polar solvent like chloroform or ether or hexane like that. That's literally the way people discovered lipids to begin with. They would take cell preparations, whatever they're dealing with, grind them up in a grinder and throw in some ether and whatever dissolved out of the cells, and they got by evaporating the ether off, literally in the subfunnel, they said, oh, that's the lipid portion. Big mixture of compounds, lots of functional groups. We're going to limit it to just these 3 right here. 3 types of lipids. The first one is the most important from a dietary point of view, and this is probably what you're thinking of when you think of fats and lipids and so forth, triacylglycerols. There's another category, though, that's important in cell membranes, definitely lipids, definitely soluble in ether and so forth, and then the steroids are kind of their own category within lipids. Okay, so we'll go through those different types. And here's what you need to know about them. Chemically, I know you're familiar with these things probably from a biology point of view and maybe dietetics as well. Here's what it looks like from a chemical point of view. So the triacylglycerols, take that word apart. Acyl is this unit right here. It's a carbonyl group with an alkyl group on it and something else. You leave that part undone. And glycerol. Okay, glycerol is a 3-carbon alcohol, and it's a 3-OH alcohol. All right. But the total thing, triacylglycerols, really has one functional group in it that is absolutely straight out of your organic class. These things are esters. An ester, hope you remember, has two components to it. One of them is a carboxylic acid, one of them is an ester, and this is in the next little diagram right here. Here's how the word comes together and how the molecules come together. Okay, they are esters. Please remember the formula for an ester. Here's the acid piece. It looks like you're starting to draw carboxylic acid. There's the carbonyl group and the alkyl group, that's the tail, and then it's like, oh, there's an alcohol piece, and that's what makes an ester. This one, naturally occurring triacylglycerols, are three of those linkages all stuck in the same molecule. This piece right here is the acid piece. It's usually along R group, which is, I'll say 12 on the short side, up to 20 carbons, and usually an even number of carbons. Okay, those 3 points, I guess, right there would be the great majority of naturally occurring triacylglycerols. And that can be a big mixture. I mean, the R group can even be this different within the same fat molecule. This one, though, this piece, no variability whatsoever. You're always stuck with this one. Glycerol is the core. It has 3 OH groups, and it's able to form 3 ester linkages. All right, in natural settings, in a plant that's making oil or an animal that's making fat, this whole thing is mediated by an enzyme. So it's the catalyst. In your organic class, the catalyst would have been sulfuric acid. And you boil, and you boil, and you go through the lab and use a SEP funnel and all kinds of messy stuff. The enzyme will catalyze this at room temperature and pressure, neutral pH, awesome catalyst. And here's the ester that you get out of it. So this would be an ester right there. That much would be an ester, and so would that. There's one, there's one, and there's one. But it's all built off of the alcohol core. The alcohol here is, there's 1, there's 2, and there's 3. So numbering, starting on the reactant side, if you're keeping track, would look like that. Those 3 carbons all in a row, the OHs strung 1 on each one of those carbons. All right, so that's what a fat molecule looks like. Dietary, or what have you. That's what we think of as dietary fats and oils. Now, the differences among them start with the R groups. They can all be different from each other, even within one molecule. Some of them, and this will be a term for you, familiar for you, I think, may be so-called saturated. And what that means is that you've got no carbon-carbon double bonds anywhere in the R group. And the kind that you've probably picked up by now are good in the diet. That's their image anyway. Some of them may be unsaturated. And those are the kinds that do have carbon-carbon double bonds in them somewhere. They are alkenes when you view them like that. All right, the terms saturated and unsaturated are still used nutritionally now. Okay, so let's talk about the details here. So I mentioned this already, and this is now broken down. The fatty acid component is this much. Okay, there's an R group. I mentioned the even number, and the naturally occurring ones are all linear. These are the natural ones. So they're pretty simple, just a whole line of carbons, a lot of them. I think I said 20 on the other page. It's low 20s. Don't worry if it's 20 or 22 or 24. So an example would be like this. 2, 4, 6, 8, 10, 12, 14, 16, 18. 2, 6, 8, 12. Sorry, I'm getting ahead of the game. It's 2, 4, 6, 8, 10, 12, 14, 16, and right there is the one I'm actually after. I'll draw it kind of bigger around it. There's carbon number 18. There's your count. Okay, this one has 18 carbons, and I drew it left to right with that numbering, but what we're going to head toward is that chemically they start their numbers with the carbonyl group. Now, this one doesn't have to be drawn that way. I just want to make sure that you know 1 to 18 is also the way that you'll see them numbered if they're numbered chemically. You're just counting them, it's all right to do it left to right. Okay, so then some information that I kind of included on the other page. All single bonds between the carbons mean saturated. The double bonds, if they are present, qualify the whole thing as unsaturated. Now, you can have more than one of these things. Many of the natural ones, we'll have like 3 to 4. We have 3 to 4 carbon-carbon double bonds. And here is maybe the main thing that gets publicity these days. The natural configuration for these alkenes is cis. And what that means is you draw the bond, the carbon-carbon bond, and you make sure that the chain goes through it and comes out on the same side as it goes in. That is a cis double bond. And that's the way they are naturally, with no exceptions as far as we know. We'll get to the trans, which are now known to be kind of bad actors. Okay, so we're going to start now with treating them chemically, not just looking at them and counting things up. We'll treat them as compounds, and we need some numbering schemes, which will look like this. I'm going to talk about a fatty acid with some numbers, and the first number is going to be your total number of carbon atoms. That much is like naming any organic molecule. You're going to name it for a parent alkane. And then the second one is going to be a number again. This one is going to be how many carbon-carbon double bonds there are in it. The more of those, the more unsaturated it is. And then there's this last little business right up here. These delta numbers. These are going to be the carbon, I want to say numbers, I think address would be a better term. Carbon addresses of the carbon-carbon bonds. Another way to think of this would be the locations of carbon-carbon bonds. Okay, so how many of you have heard of talk of omega-3 fatty acids? Is that discussed these days? We're not quite there, not quite ready to talk about it, but omega-3 is a way of talking about not the whole molecule. It's a way of talking about just the location of the double bond. Okay, that's what we're headed for right here. We'll do some standard numbering right here to begin with, and then we'll get to the omega. The omega is probably what you're more familiar with, but let's do the standard. Okay, now this time I've got them written from left to right, so your urge to number them will be properly lined up with going left to right. So here's number 1, the carbonyl group. We'll call that number 1 on all of them. And how many have we got here? Well, I don't want to write these on. 1, 2, 3, 5, 7, 9, 11, 13, 15, and 16. So let's take that one as kind of our practice case to begin with. And here are the standard symbols. We're not into omega yet, but we'll get there. So your standard symbol would say how many carbons to begin with? Well, 16. I'll label this a little better in just a minute. Actually, let me put that right there. How many carbons? And then the number of carbon-carbon double bonds is exactly what? Number of alkenes? None. Okay, that says that you here have no carbon-carbon double bonds. And that means that up here you've got no delta. There's nothing to write. So as long as you write 16:0, you are considered to be informed, avant-garde, and cool in talking about fatty acids. You are in the know. Now it's not as simple when you get to this next one though. I think it should have the same number. Let me count them. So here we're up to 1, 3, 5, 7. Double bond starts at 9. And goes to 10, then 11, there'd be 15, 17, this one's a little longer. Okay, that one's 18. If I did my count right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Whoops, I jumped too quick. That should be 13. That would make this one 15, and 16, sorry. Okay, yeah, they're the same length, but look at the difference between the two of them. This one has a double bond, and look what I did not draw. I think the common urge is to say, well, I've got a double bond at number 9, so I just draw my chain right here, and then I go to number 9 and go, draw on my double bond, and I'm done. Except that if it's a cis double bond, you must put a kink in the chain. That's what all naturally occurring fatty acids made in plants look like. It has to be cis. Okay, so you're going to go here and you're going to say, what's the symbol for this one? Well, still 16 carbons. That starts out the same. And now you need to give the number address. But first of all, talk about how many double bonds there are in it. That's 1. So here's the difference here. You have 1 of these things to go with that number. And now you need, since you said that there's 1 in there, you better be prepared to tell where it is. And it starts at number 9. So that spot right there, that number, that's the location of your carbon-carbon double bond. Just moving it by 1 carbon turns it into a different molecule. Right? You know what constitutional isomers look like. It does matter in lipids. And here's another one right here. So let's get a count on this one. 2, 3, 5, 7, 8. Okay, that double bond starts at 9 as well. 10, 11, 12, 13, 14, 15, 17. This one is a little bigger. Okay, that's what you're looking at. And so, of course, you'd have to start this one by saying the symbol for it is 18. I'm talking about an 18 carbon fatty acid. There are an awful lot of those, so you narrow things down by saying, well, how many double bonds are we talking about? 2. So this one is more unsaturated than the first one. And it has 2 different locations where those double bonds start. One of them looks like it's at 9, and the other one looks like it's at 12. So that's all the symbol you need for this one. There's a trend here. This one right here, the first one, is totally saturated. This one is unsaturated. and this one is polyunsaturated. Okay, are you still given the information that these are nutritionally preferable to these? Is that still the model? Okay, yeah. So, in other words, the ones that work best in your diet are the ones that have the most kinks in their chains, and that's because you put together something like this. That's kind of a challenge. You first of all got to emulsify it in your gut. Well, same here for that matter, but there are enzymes that are more prone to going after this. Okay, there's an alternate symbol which I mentioned earlier. It's the omega number. It starts from the other end, from the last carbon in the chain, and this is compared to away from the carbonyl. So this isn't your usual numbering that would have come out of an organic class, but it does help for the disciplines that need to talk about how far a double bond, how much an unsaturation is, away from the very terminal end. The very fact that we're talking now about this says, well, you need to put some designations on it somehow. So here's omega, that's the last one. Here's omega 1, here's omega 2, and here's omega 3. You may see numbers like that. This one would technically be omega 2 because it starts on the omega 2 carbon and stretches to number 3. The famous ones, omega 3, would start 3 and go to here. So this one, this is an omega 2 fatty acid. Okay. Any of those are correct, and you use them according to the discipline that you're talking to, whether it's biologists, nutritionists, chemists, it's all part of the same thing. Okay, what time we got? I think we better call it done there so we don't jump off into something we can't finish conveniently. Okay, yeah, that's the end for today, and I'll plan to see you on Wednesday.